Ladies and gentlemen, thank you for standing by, and welcome to the Trillium Therapeutics conference call. At this time, all participants are in a listen-only mode. After the speaker's presentation, there will be a question-and-answer session. To ask a question during the session, you will need to press star 1 on your telephone. If you require any further assistance, please press star 0. I would now like to hand the conference over to your speaker today, Jan Skavarka. Please go ahead.
Thank you, Josh. Good evening. First of all, I'd like to thank everyone for dialing in at such a short notice. As is customary, I will be making forward-looking statements this evening, so let me point you to our forward-looking statement disclaimer, which you can find in the updated corporate presentation on our website. So it is with a great pleasure that we are hosting this call on the heels of two substantial announcements we have made earlier today. First and foremost, we have provided a data update for both our molecules, TTI-621 and 622. In the update, we are reporting substantial monotherapy activity with 622, with a strong safety profile and further dose escalation from 8 to 12 milligrams per kilogram. Similarly, with TTI-621, we continue seeing a signal of single-agent activity with acceptable safety profile. Most importantly, we have not seen any thrombocytopenia DLTs, and we are further escalating the dose from 1.4 to 2.0 milligrams per kilogram, as we previously announced. Second, we announced a $25 million equity investment from Pfizer, and Pfizer Oncology Chief Scientific Officer, Dr. Jeff Settleman, to be joining our scientific advisory board that we are currently forming and will be announced later this year. Now, before I dive into details of the two announcements, I'd like to provide some context. These are very exciting times for both the CD47 field and Trillium Therapeutics. The CD47 field is generating promising clinical data in a range of heme malignancies, as well as preclinical and emerging clinical evidence in solid tumors. This encouraging data is attracting substantial interest from smart healthcare investors and strategic partners. We are seeing healthy competition in the field from mostly biotechnology companies, but also from big pharma that is gradually entering the space. The field is exploring a variety of different approaches with monoclonal antibodies, fusion proteins, or bispecifics. We are testing different molecule constructs involving different Fc isotypes, using different molecule affinity models, and so on. And in this rapidly evolving field, we believe that Trillium is well positioned to develop a true best-in-class molecule. We have two shots on goal, SERP alpha fusion proteins built on IgG1 and IgG4 scaffolds, respectively. The molecules are testing two different hypotheses. The 621 is utilizing a strong EAT signal via an IgG1 Fc and with a relatively low dose, currently dosing at 2 milligrams per kilogram, while 622 is using a moderate EAT signal via IgG4 Fc and a substantially higher dose, now dosing at 12 milligrams per kilogram. Both molecules are highly differentiated in the field, specifically across three dimensions. Number one, monotherapy activity. Number two, lack of red blood cell binding, which has several benefits, including no anemia effect or no wasting of the drug, no waste of the drug in the RBC sink, among others. And third, molecular weight that is one half of that of monoclonal antibodies, which gives us a potential advantage in the solid tumors. But the key differentiating factor we are most excited about is the monotherapy activity of both 621 and 622. And it is not because we envision our molecules to be used as single agents in the clinic. On the contrary, we do expect those molecules to be used predominantly in combination with other agents. Rather, it is because monotherapy activity gives us confidence in the bioactivity of our molecules and in our data. The data we show are attributed to our molecules and not to a combination agent. But there's also a second equally important advantage of having single-agent activity. It is to provide a stronger and much more robust foundation for combination therapies, including PD-1 drugs, which do not carry an EAT signal, which is required for inducing macrophage phagocytosis. And the goal of combining CD47 with PD-1 agents is to potentially generate deep fast and durable immunologic responses by engaging both arms of the immune system. Now, this is, of course, in the realm of hypotheses today, but if successful, such approach could change a decades-long paradigm in oncology, with the ultimate aspiration being to challenge chemotherapy as a first-line attack in a variety of cancers. So this is perhaps a bit longer introduction than you might have expected, but I wanted to provide some context for the most important update we have today. It is the emerging single-agent activity of TTI-622. Now, of course, in addition to the previously observed and now continued observation of single-agent activity of 621. So with that said, let me talk first about the clinical updates, then about the Pfizer investment, and finally we'll open it up for Q&A. So let me start with TTI-622. As a reminder, 622 is a SIRP alpha IgG4 fusion protein. It is currently being evaluated in a phase 1 single-agent study in relapsed refractory lymphoma patients. We are using a 3+3 escalation schema, weekly infusions, and treat until progression. We have enrolled 26 patients across 6 cohorts with the highest dose of 8 milligrams per kilogram to date. My update has 3 parts. First, activity, second, safety, and third, pharmacokinetics. As a reminder, the last update we provided was at ASCO on May 29, so about 3 months ago, a little bit more than 3 months. So first, in terms of activity, you may remember that at ASCO we reported 2 responses, a complete response and a partial response. Since then, we have observed 4 more objective responses. So today, we have 6 out of 18 responses, or 33% objective response rate, at the combined dose levels from 0.8 to 8.0 milligram per kilogram, which is cohorts 3 to 6. And this is including 3 out of 6, or 50% ORR at the highest dose of 8 milligram per kilogram. But the true reason we are excited about this monotherapy activity is not just the simple statistic of 6 out of 18, but rather it is the holistic picture of these responses. Specifically, number 1, it's the broad therapeutic window with responses at 4 adjacent dose levels from 0.8 to 8 milligram per kilogram. Second, a hint of dose-response relationship, as we are observing the highest response rate at the highest dose level. Number 3, the responses are observed across different indications, lymphoma indications. Number 4, it is the highly relapsed refractory nature of responders with 3 to 9 prior systemic therapies. Number 5, responses are including a complete response and a near complete response in 1 patient. And number 6, it is the fast onset of action. And this is it's quite remarkable. All 6 responses were observed at the very first response assessment at week 8. As a side note, one active patient in the last cohort at 8 milligram per kilogram has yet to have response assessment, so the 50% or 3 out of 6 response rate for the 8 milligram per kilogram cohort will change by that 1 patient response. Now, more importantly, though, let me put this monotherapy activity data in context of some of our CD47 competitor data from their combination studies with rituximab, with the obvious caveat that we do not have a direct head-to-head comparison. So just to reiterate, with single agent TTI-622, we observed 33% ORR at the combined doses of 0.8 to 8 milligram per kilogram, including 50% at 8 milligram per kilogram in highly pretreated patients across a range of lymphoma indications. Magrolimab showed 36% ORR in relapsed refractory DLBCL patients, and it was at magrolimab's full dose of 30 milligram per kilogram in combination with rituximab. Now, similarly, also ALX148 showed 55% ORR. This was at their full dose of 15 milligram per kilogram, and also in combination with rituximab. So in summary, we feel that in lymphoma monotherapy response data with TTI-622 and the doses we have studied today is roughly comparable to the DLBCL response data for other CD47 blockers when they are used in combination with rituximab and at their full doses. Now, second, the safety profile of TTI-622 continues to be very strong. At 8 milligrams per kilogram, we observed one dose-limited toxicity of grade 4 thrombocytopenia with prophylactic transfusion in one patient. However, after the event, the patient resumed study at 4 milligrams per kilogram. This DLT event has been the only related severe adverse event we have observed to date. And most importantly, we have completed safety assessment of the 8 milligram per kilogram cohort and are now escalating to 12 milligram per kilogram dose. Now, finally, third, we are seeing favorable pharmacokinetic profile with the dose-dependent increases in serum exposure, which supports further dose escalation beyond 8 milligram per kilogram. Now, let me switch to TTI-621, our SERP alpha IgG1 fusion protein. Just for clarity, the IgG scaffold is the only difference between 621 and 622. And as I mentioned before, with 621, we are testing a hypothesis of combining a strong EAT signal with a relatively low dose of the molecule versus a moderate EAT signal with a higher dose of the 622 molecule. 621, as you may remember, 621 has been dosed in over 200 patients to date, most at an initial maximum tolerated dose of 0.2 milligram per kilogram, determined under initial DLT criteria for thrombocytopenia as a grade 4 of any duration, so no time qualifier. After firmly establishing that thrombocytopenia was a transient event, the DLT was revised to the commonly used criterion of grade 4, lasting 72 hours or longer, which allowed us to test doses of 0.5 milligram per kilogram and higher in the current part 4 of the study. So more specifically, in part 4, we are now testing TTI-621 in relapsed or refractory CTCL patients at pre-specified dose levels of 0.5, 0.7, 1.0, and 1.4 milligram per kilogram, with the protocol allowing for flexibility to study higher doses per safety review committee discretion. So as for activity of TTI-621 in part 4, we continue to see a signal of monotherapy activity at the 1.0 milligram per kilogram level. We observed 1 out of 6 patients, or 17%, achieving a partial response, and 1 additional patient achieving skin complete response, albeit the overall assessment of that patient was stable disease. 2 patients in the cohort were bridged to bone marrow transplant. At the 0.7 milligram per kilogram cohort, we observed 1 blood complete response. Overall assessment was stable disease in 3 patients. Furthermore, we are also seeing a dose-dependent improvement in skin disease score as measured by MSLAT, or it stands for Modified Severity Weighted Assessment Tool. And finally, the activity data for the 1.4 milligram per kilogram cohort is not yet available at this point in time. From a safety perspective, TTI-621 has been well tolerated. The only grade 3 or higher related adverse events were infusion-related reactions and thrombocytopenia, each with 13% incidence. We observed 1 infusion-related reaction, DLT, at 1.0 milligram per kilogram. In general, infusion-related reactions occur almost exclusively during first infusions, and they are managed by prolonged infusion time and steroid pre-medication. Critically, though, we have not observed any thrombocytopenia-related DLTs in part 4. The relatively well-tolerated profile of 621 allows us now to dose at 2 milligram per kilogram, which is higher than the highest pre-specified dose in the protocol. Now, stepping back, we are really excited about having 2 attractive shots on goal now also with 622 reporting substantial monotherapy activity. We expect to declare maximum tolerated doses or recommended phase 2 doses for both molecules, either towards the end of this year or in the first half of 2021. This is of course subject to any potential coronavirus-related delays or uncertainties of those selection efforts. Our clinical development priorities going forward will include 4 heme malignancies, specifically AML-MDS, multiple myeloma, DLBCL, PTCL, as well as solid tumors. Solid tumors will be a very important part of our strategy going forward. We are in the process of defining our specific indication priorities, such as patient settings, intervention, for example, which combination agents, regulatory path, and so on. And we will announce a clinical development plan around the time we declare 621 or 622 maximum tolerated doses or recommended phase 2 doses. So with this clinical update, let me now move to the second announcement of today, the Pfizer equity investment. Pfizer is investing $25 million in common equity at $10.88 per share, which is a 15% premium to today's closing price of $9.46 per share. This is a straight equity investment, and Trillium is retaining all product and regional rights. As part of the transaction, Pfizer Oncology Chief Scientific Officer Dr. Jeff Settleman has agreed to join our scientific advisory board that is currently being formed and will be announced later this year. I am particularly pleased about this, as Dr. Settleman is one of the leading cancer therapeutics experts. The SAB, again, is in final formation stages, and as I mentioned, will be announced later this year. The SAB will help guide our overall scientific and clinical priorities, and we believe that Dr. Settleman will be able to substantially contribute to its discussions. So overall, very pleased with Pfizer taking interest in our CD47 program, and we are looking forward to working with Dr. Settleman within our SAB. So this completes the first part of this call, and we will now move to the Q&A session. Joining me for the Q&A session will be James Parsons, our Chief Financial Officer, Dr. Bob Uger, our Chief Scientific Officer, and Dr. Yating Xu, our Chief Medical Officer. Josh, we can kick off the Q&A session.
At this time, I would like to remind everyone, in order to ask a question, please press star, then the number 1 on your telephone keypad. If you would like to withdraw your question, please press the pound key. We'll pause briefly to compile any questions. As a reminder, please press star, then the number 1 on your telephone keypad if you would like to ask a question. I currently show no telephonic questions at this time. Again, as a final reminder, if you would like to ask a question, please press star, then the number 1 on your telephone keypad. There are currently no telephonic questions, Jan.
Okay, I see we have quite a long list of participants, so maybe let's give it a little bit more time. So I just got, Josh, I just got an email, actually several emails, trying to ask a question, but not, I am pressing star 1, not getting through.
1 second, let me try this and I'll see if it's... And we do have some questions. Your first question comes from Ren Benjamin with JMP Securities. Please go ahead.
Hey, good afternoon. Thanks for taking the questions. Glad to make it through. Congratulations, Jan, on this really nice update. Maybe just a couple of quick ones. Thanks. Maybe just a couple of quick ones. Can you talk a little bit about the durability of the responses that you're seeing right now? I imagine that the PRs that you're reporting on have gone through the customary sort of at least 2 scans, but how long are these turning out? And then also whether or not we've seen any activity. You mentioned several tumor types that we were going to be that you're going to be evaluating, multiple myeloma being one of them. Have we seen any activity in multiple myeloma? And if I can leave you with one final question just to think about or to address, how do we think about moving both these molecules forward? They do have differentiating aspects. How should we think about which one goes with which combination and with which indications?
Okay, good. So let me recap. So the first question was durability.
Yes.
Second was, sorry, what was the second question?
Sorry, just regarding the indications you guys had mentioned,
multiple myeloma, right? Yeah.
Multiple myeloma, yeah.
Yeah, okay. Yeah, okay. So durability is just way too early to talk about, Ren, especially the 8 milligram per kilogram cohort. All these responses, you may remember the last update we had at ASCO of May 29, and so we just had 4 responses coming in over the past few months. So it's too early to talk about durability. The one patient, the one CR, we reported it as the very first response. The patient is still continuing on treatment now for 16 months. But more importantly, it's just way too early to talk about durability at this point. Second, multiple myeloma. Let me put it on hold for a second. I will ask Bob Huger to address this question. And in terms of the 2 molecules, we don't know at this point yet. There are several scenarios. In one scenario, if the clinical profiles of the 2 molecules sufficiently emerge out of the dose escalation studies, we might be able to declare a winner. But there's also a different scenario under which both molecules are strong, and in which case we continue with both molecules in program 2. And then again, we have 2 sort of sub options. And one sub option is pick indications or combinations, so we do not do parallel. We don't have parallel efforts. Or it is also possible that we may study the 2 drugs head to head in one or a few indications. So, but importantly, it's too early to tell. We'll see how the clinical profiles of the 2 molecules evolve during the dose escalation studies, and then we will make the decision after. As far as for multiple myeloma, Bob, so we do not have clinical evidence in multiple myeloma. However, we have preclinical evidence in multiple myeloma, which we also published. So, Bob, can you please address the question?
Yeah, sure. So our interest in multiple myeloma comes from several aspects. The first is just generally in the literature, we know that CD47 is overexpressed in multiple myeloma. We know that overexpression correlates with worse prognosis in that disease. But more specifically, I think we have pretty robust preclinical data in multiple myeloma. In vitro, we know that we can get macrophage with our drugs. We can get macrophages to very efficiently phagocytose and kill multiple myeloma cells. And we also have in vivo data in xenograft models, both as a single agent as well as in combination with proteasome inhibitors as well as with daratumumab. So like I said, I think a fairly robust preclinical dossier in this disease that gives us a lot of enthusiasm for treating multiple myeloma patients.
Terrific. Great. Thanks for taking the questions, and I'll jump back into queue.
A pleasure. Thank you, Ren.
Your next question comes from David Martin with Bloom Burton. Please go ahead.
Hi there, guys. Congratulations. My first question is, do you have any thoughts why you're getting such high monotherapy response rates with TTI 622? We kind of expected it with 621, but wouldn't you expect that 622 would be more in line with the IgG4 antibodies?
Sure. Bob, why don't you take this question and explain the two hypotheses we are testing?
Yeah, I mean, it's so we, of course, with our 621 and 622 programs, the two hypotheses that Jan is alluding to are, this idea that with 621 we have a molecule with a powerful EAT signal that's given at relatively modest doses, and then 622 is a molecule that carries a more modest EAT signal but is given with the higher exposures. And so whether that, ultimately that's sort of the hypothesis that we're trying to test between these two drugs. To the question about, why we're seeing greater monotherapy activity, I mean, we certainly expected at least some monotherapy activity with 622 because it does carry its own EAT signal. But to the extent that we're seeing it, that's an area of active investigation. And, I think it's safe to say, there's a lot of different CD47 blockers out there. We're learning as the clinical data come in that not all of these blockers, even among the same isotype, are created equal. And if you look within the IgG4 class, we're seeing that, I think, with 622 and magrolimab and Celgene's antibody. So, we have a number of hypotheses, but they're at the hypothesis stage right now, and they are currently being tested.
Okay.
So I think the key point is not all these IgG4s are created equal, and I'm sure the Celgene people are also scratching their heads. It's how come their molecule, the CC90002, did not produce any monotherapy activity. On the other hand, we have magrolimab, which actually did show some monotherapy activity in AML, MDS, and some signal in solid, weak signal, but nevertheless showed a monotherapy signal in solid tumors as well.
Okay, we'll stay tuned. Second question is, in the 621 trial, you had that patient with the overall stable disease, but the skin complete response. I'm wondering how clinically relevant is that? And then the two patients that bridged to transplant, was it that patient and the other one who achieved the partial response?
Okay, Ping, can you please address the question?
So for the first patient who achieved skin CR, the patient does have the disease in lymph nodes as well. And obviously, when we observe the skin CR, the lymph nodes response is somewhat delayed. So we will have to continue to sort of observe. Clinically, it's significant to see the complete resolution of the skin lesion. Obviously, that's the major sort of disease for CTCL patient there. The other two patients in the 1 mg per kg cohort transitioned to transplant. Their response, definitely we've seen the skin improvement, and think the mSWAT is close to reaching the PR, but not quite. But these patients had a donor available, and obviously these patients are at young age and eligible for the allogeneic transplant. So obviously we have to consider the best interest for the patient, and so the patient was transitioned to transplant. Now one could sort of speculate if these patients continue the treatment on our study drug, whether or not the patient will be achieving objective response. There's definitely a possibility there.
Okay, so at the 1 milligram dose, there was one that achieved a PR, one that had a skin CR, and one that was a near PR. Is that correct? And that was 6 patients in that cohort?
Yeah, the two patients transitioned to transplant both had a considerable skin lesion improvement, but not quite objective PR.
Okay, thanks. That's it for me.
Very good. Thank you, Dave.
Your next question comes from Sumit Roy with Jones Trading. Please go ahead.
Hello, everyone, and congratulations on this very robust monotherapy data. I got a couple of questions. One is at 8 milligram dose with 622, the 3 PRs we are seeing, could you say they are all from DLBCL or spread over TCL and follicular lymphoma, which indications they have? And second is...
yeah.
Very good.
Mm-hmm.
Go ahead.
No, go ahead, Sherman.
So the second question is going back to the ASCO data when you presented receptor occupancy at a lower dose of 622 around 70%. So should we start thinking like 8 or 12 milligram could be the RP2D given it has the size of magrolimab, so instead of 30 it could be 12 the max? And could you tell us like what's the MTD criteria? What would make you finally determine this is the MTD and we'll decide on the RP2D?
Yeah, okay. All right, so for your first question, at 8 milligram per kilogram, the 3 responders, 2 responders had CTCL with the large cell transformation, and 1 patient had follicular lymphoma. And as for the recommended phase 2 dose or maximum tolerated dose, I mean, there's obviously we may hit an MTD, so in that case, decision would be simpler. Or we may go the recommended phase 2 dose route similar as magrolimab or ALX148 when we would declare the dose based on the holistic picture of PK and RO data. I don't know, Bob, if you want to add anything to that.
No, I certainly agree with that, and we're characterizing drug levels and receptor occupancy levels. We have seen that those the occupancy levels are increasing, and in particular, sort of the durability or the sustainability of the RO is increasing as we move up from 8 mg per kg, and we're not, we haven't yet seen a plateauing effect of the exposure relationship. So. We continue to monitor those parameters, and those will certainly factor into our dose determination.
Thank you so much, and congratulations again.
Thank you so much.
Your next question comes from Boris Peaker with Cowen. Please go ahead.
Good afternoon, and I'd like to add my congratulations on the great data. Just 2 questions for me. First, on the grade 4 thrombocytopenia for 622, can you just comment how that was defined? Was it similar to 621 where it needs to be at this event rate for 3 days before it's recorded, or was it just a single trigger event? And my second question is on kind of the path forward. Do you have a sense of how many patients worth of data you think you'd need to see before you could start the expansion studies and really deciding on the indications to pursue for, I guess, either drug?
Okay, Yaping, why don't you go ahead with the first question?
Yeah, so the grade 4 thrombocytopenia event occurred in 622. With regard to the DLT criteria, there is a consistency between how we define DLT regarding the thrombocytopenia. So there's several different sort of parameters. One is grade 4 lasting for 72 hours. That's one. Or the platelet decrease to equal or less to 10,000. The third is if the thrombocytopenia in investigators' opinion require transfusion, that's another criteria that we can use to define DLT. So in this particular case, the grade 4 thrombocytopenia occurred. there's no sort of clinically meaningful symptoms. There's no any evidence of any hemorrhagical event occurring. So clinically, there's no sort of indication for platelet transfusion. And also, according to ESCO practice guideline, the platelet count the patient had does not meet the criteria for prophylactic platelet transfusion. However, you all know we're under the COVID-19 situation, and the treating physician is making effort trying to avoid any unnecessary hospital stay. So in this case, it's the investigator's discretion to give the patient prophylactic platelet transfusion, and because of that, it meets the sort of a third parameter for the DLT criteria. And we also support of that sort of action because this was the second patient who dosed at the 8 mg per kg. So definitely we want to exercise some caution at a time. We think calling this DLT will allow us to expand further up the cohort, enroll more patients into the 8 mg per kg cohort. So now we feel much more comfortable with the 6 DLT evaluable patient, this dose level there. This is the one single incident of a grade 4 that occurring at this dose level there. Particularly with regard to this patient, after the platelet transfusion, platelet counts coming back, and the patient was no longer grade 4 the second day, and in a week the patient restart the study treatment. Obviously, because of DLT, is at a lower dose at 4 mg per kg, but the patient had been doing very well.
This was also, Boris, this was somewhat idiosyncratic safety event. We actually did include a platelet level chart on page 26 in our corporate deck, and then you will see if you are at your computer, and you could see that in cohort 6 there was one patient which was a little bit sort of out of the range. So that was the reason why we included to show it was the idiosyncratic nature of this DLT event. So it is, but to your question, how many patients do we need to see to move forward to phase 2? So this is a 3+3 dose escalation schema. So the simple answer is 6. Having said that, we actually have quite a substantial backlog of patients. The PIs are very excited about enrolling their patients. So we are evaluating different options how to enroll more patients, but it's still TBD.
Great. Well, thank you very much for taking my questions, and once again, congratulations on the data update and Pfizer's investment too. Thank you.
Oh yeah, thanks, Boris.
Your next question comes from Robin Garner with Craig-Hallum. Please go ahead.
Hi, everybody. Congratulations again on the update. Just wondering if you could provide any additional information about your strategy in solid tumor, whether there's data that you can share with us on the call today, or just any further thoughts on it.
Yeah. Okay. Thank you, Robin. And at this point, we are the gatekeeping factor for us is to complete those escalation studies and to identify maximum tolerated dose or recommended phase 2 dose. Now, in parallel, we are articulating our strategic priorities in both heme malignancies and in solid tumors. We are in final stages of formalizing and creating our scientific advisory board, of which Jeff Settleman from Pfizer will be a member of. The SAB will be providing an overall scientific and clinical guidance for our strategic direction, and then underneath we have key opinion leader panels, which are indication specific. So the strategic planning process is very much work in process, and we are not yet ready to announce the full detailed clinical development plan. But again, it's solid tumors will be a very important part of the strategy going forward. Pfizer has a great interest in solid tumors as well with CD47, and we just need a bit more time to define the strategy with the SAB.
Okay, thank you very much for that. And also, just regarding the Pfizer investment, a competitor program recently announced a significant strategic investment with a strong upfront. And curious if what were your thoughts in terms of the structure of this Pfizer investment versus how other competitor programs have been taking on strategic partners?
Right. Yeah. Thank you. So the IMAB deal and what we announced today with Pfizer are two very different things. IMAB basically sold their molecule to AbbVie, or sold their worldwide rights, except for China. They sold the rights for $180 million or $200 million upfront, plus milestones, $1.7 billion milestones and royalties. It's very different what we have. We do not have really a deal. This is what we have is a straight equity investment, $25 million at a certain premium, where their chief scientific officer, oncology CSO, is joining our SAB, and we are retaining full rights, full product and regional rights. And actually, I also received via email a separate question, which relates to your question, Robin, as well. The question reads, regarding Pfizer, since they now have a seat at the table, do they have any rights of first refusal in either partnership or a takeout? And the answer is no. So it was very important to us that we are, at this point, we are retaining full rights to our both programs, and we are keeping our options open for any partnerships or other strategic considerations down the road. And we believe this way we are creating more value for our shareholders and finding the right path to bring the molecules to market for patients.
Okay, thank you very much. That's it for me for today.
You're welcome, Robin.
Your next question comes from Swayampakula Ramakanth from H.C. Wainwright. Please go ahead.
Thank you. Thanks, Jan, for taking my questions. A lot of them have been answered. A couple of quick ones. For the 621 study, we know that this has been going on for a bit of time. So what gives you confidence that you will get to MTD or a recommended phase 2 dose by year-end or early 21?
Yeah, okay. Yaping, do you want to address this?
Yeah, so obviously we are at the dose level that's already beyond original plan, and we will see whether or not any safety signal will help us to have a pause or determine the dose. And on the other hand, the purpose of a dose escalation is really trying to see whether or not increased dose and improved dose exposure could greatly enhance the efficacy signal. And obviously we have the data up to 1.0 mg per kg from the efficacy side. We would like to continue to evaluate the clinical activity at 1.4 and 2. So we will consider both from the safety end and also whether or not there is a substantial gain from the clinical activity by dosing up, and we will make a decision when we have those data available.
Okay, thank you. And then on 622, as you stated, monotherapy is good, and you have positive data with good efficacy. But it seems like combination therapy is the way to go with these molecules. If when you come to that decision, especially when going to the next stage of development, are there any specific checkpoint inhibitors that you are thinking of, or outside of checkpoint inhibitors that could be potential combination therapies to go along with 622?
Bob, why don't you go ahead, please?
Sure. So in the checkpoint inhibition, that's definitely an area of interest for us with anti-PD-1. I think that from a scientific point of view makes a lot of sense. Blockade of checkpoints for both the innate and adaptive immune system. This is obviously not a completely novel concept. Others have already begun those types of studies. So I think, the CD47 field is moving in that direction. Beyond checkpoint inhibitors, there definitely are other molecules, other classes of combo agents that are under consideration. I can go through a few of those just very briefly. Azacitidine is one, obviously, given the clinical validation that we've seen in AML and MDS. Antibodies such as daratumumab or cetuximab in the solid tumor world. In other spaces, I mentioned in multiple myeloma, proteasome inhibitors is an area that we have preclinical data supporting, as well as pralatrexate. We consider other chemotherapies as well. So there really is quite a number of different combinations, and these will, some are obviously more suitable for certain indications as compared to others.
Thank you, Bob. Thanks, Jan. I'll talk to you soon.
Thank you, RK. Have a nice evening.
There are no further questions at this time. I'll turn the call back to Jan Skabarka for any closing remarks.
Great. Well, thank you again, everybody, for calling in at the short notice and at the late hour. Again, we're very excited about, most importantly, of course, about the data, but also about the Pfizer investment and Jeff Settleman joining the SAB. And one final comment is we will provide more updates on our data at ASH. I think actually I received some emails. How should we think about ASH now? Did you break the embargo? So for situations like this, when there is an investment and we have an obligation due to SEC rules to disclose the data, and we receive an exemption from ASH from their embargo. So again, we'll have more data at ASH for you. So thank you again, and looking forward to talking to everybody in the future.